The last kind of tissue we're going to talk about is vascular tissue. Actually, there's another one we're going to talk about, but it's only for secondary growth. But the last primary growth tissue is vascular tissue. And there's two basic types. There's xylem and phloem. Xylem is for transporting water and nutrients from the roots and from the soil up to the rest of the plant, mostly. And phloem is for transporting photosynthase or materials, sugars mainly made in photosynthesis, down from the leaves to the rest of the plant. So both of them are transport tissues, and generally speaking, they're found somewhat in the middle of the plant. There's different arrangements for different parts of the plant, and we will talk about that in a little bit. So I'm going to go over the different cell types in each one of these. So let's first start by the cell types that are found in xylem. Now, in xylem, we have vessel members and tracheids are the main cell types. Now, there are other cell types that can be found in both xylem and phloem are parenchyma and sclerenchyma, and they would just be kind of in between or surrounding the actual vessel elements and tracheids that would be found together. Now, most higher level plants have both. Some lower level plants only have tracheids, so they're considered less advanced, and they are narrower, and so the plant would necessarily be smaller, not needing to transport as much water, etc. And that's another reason why they're considered less advanced. But anyway, in most higher level plants we have both, and they exist kind of as a unit. In other words, they're making tubes that are going to be next to each other, and they're going to be connected in one big unit of many of these tubes of both types. Okay, now in the vessel elements, they have what they call a perforation plate at the end of each one of the cells, and this allows the water to flow through the cell wall continuum. Now what happens to create that is that the cell will start off as a regular cell, and then it will die by creating it, and also create a secondary cell wall. And as it dies, the cytoplasm and the vacuole and all the organelles and everything disappear, leaving a hollowness inside of the cell. Now that hollowness allows then for the water, it basically then that creates a tube through which water can flow, and that happens in the tracheids as well. There's also little holes in the side of each cell called a pit, and those pits allow water to go sideways through the, so like say for example something was blocking this perforation plate, then the water could go sideways and go in the neighboring tube, so that water being sucked up the plant will actually, can go in many different ones of these tubes that are all in a unit together. The tracheids also have that. Now some features of the xylem cells are that they are all dead at maturity, and they are functional only when they're newly dead. Now in the primary plant growth that we've been talking about so far, everything would be functional, all the xylem cells would be functional. But later on, when we talk about secondary growth, you're going to see that there is some xylem that's a little bit older, like last year's, and last year's xylem would not be functional. Only this year's newly dead xylem would be functional. The reason is it ends up filling up with things like it'll fill up with either debris or resins or something like that that will kind of block the last year's xylem. So they all have secondary cell walls. What's unusual about these secondary cell walls is that they are in a helix shape. In other words, they spiral down the cell, or they are in annular shape, that is in little groups in rings that go down. And in the lab, I will show you some models of this, and so you can kind of see what it looks like. Okay. The other kind of cells are in the phloem. I'm sorry. The other kind of tissue is phloem, and the cell types that are found in the phloem are sieve tube elements and companion cells. They also have, just like in the xylem, parenchyma and sclerenchyma, kind of surrounding, again, a group of sieve tube elements and companion cells. Now, in this case, the sieve tube element has to be alive at maturity. Now the reason for this is that it has to be able to transport sugars into this lumen, or that is the open space within the sieve tube element. So in order to do that, you have to have membrane, and you also have to have some nuclear function. You have to be able to have ATP and use that ATP to transport the sugars across that membrane. We haven't really talked about that much at all yet. We will in the next unit, but I'm just telling you this in advance. So these cells have to have the ability to function. So they have to be alive. Now these guys are barely alive. They have cytoplasm that's kind of just on the outside edge. If it was 3D, this would be going all the way around. I tried to draw it up here. And then that means they also have to have a neighboring cell that has a nucleus that is doing the nuclear support for both cells. Okay, so literally speaking, these cells are formed together. One contains the nucleus, the other one does not, and they always exist side by side. The companion cell is the one with the nucleus, and the sieve tube element is the one that is basically hollow, allowing materials to flow through it. All right, so the last kind of tissue I'll just tell you about, and then I'll show you some overheads. The last kind of tissue, this is a secondary tissue, is called the periderm, and it's going to end up replacing the outside part of the plant, which you'll find out as we go through the tissues. It's called epiderm and cortex. Later on, write that down, and later on that will make more sense. And it will create 3 types of cells, or you could even say these are tissues: cork, cork cambium, and pheloderm. These exist as layers on the outside. The cork is like the dead layer of cells. Those cells have secondary cell walls. It's completely dead. Cork cambium is a living part. It's actually a meristem that is going to keep replacing or keep making cork towards the outside, and pheloderm, keeping the pheloderm maintained towards the inside. And the pheloderm is one little layer of cells that's right inside the cork cambium that is next to the actual living part of the tree. And we'll talk more about this periderm later, but I just wanted to mention it as a tissue type at this point, okay? So the first thing, though, let's go through these overheads a little bit so you can look at them a little closer. So these are actual pictures of the xylem and the tracheas, and they're showing you, this is of course a drawing kind of like mine, showing you all the features that I was talking about, but these are actual pictures. So you can see there's a lot of pits on the side, which, you know, are kind of hard to draw, and the tracheas, you can see how narrow they are. And as they exist all together, you can maybe see where there might be sclerenchyma, which is really supportive cells, and then also perhaps parenchyma in there, replacing anything that actually dies off, okay? Oops, sorry. Another one, another overhead here is, this is the phloem. So this is showing you the phloem cells. This would be the sieve tube element with the sieve tube plates. And then right next to it is going to be the companion cell. So here's what it looks like in reality. Okay, so you can kind of see a little bit how the sieve tube element is hollow in the middle with the cytoplasm on the side, and then the companion cell over here. I'm sorry, my dog is squeaking. There's my dog. Hello. All right. And then I wanted to show you, these are like, this is like the formation of these cells. So here is a formation of a xylem cell. So as it starts off as an initial, and then it becomes a derivative, it's going to have, you know, normally a nucleus and a vacuole, right? And then it's going to start forming those secondary cell walls, which will be, in this case, it's showing you kind of a helical, that is, it's winding around like that. It's kind of hard to see. And then the vacuole will go away and the nucleus will go away, leaving behind the hollow cell. So that's kind of like the derivation of that kind of cell. And then lastly, the derivation of the, this is like the companion cell. So here's the initial phloem cell, and it had one nucleus that will divide. And then as that nucleus divides, it's going to create a wall between the two nuclei. And then This part leaves the cytoplasm behind, but the vacuole disappears. So it'll have cytoplasm, and there's the companion cell. So that's like how they are derived there. Just to kind of mention, other cell types like fibers or sclerenchyma would do the same thing. So this is an initial, oops, this is upside down. Maybe I'll get this right someday. Okay, so this is an initial, and these were the xylem and the phloem. All right, but a fiber would be like this, so it's going to start off with a nuclei and then that dies. Remember, fibers or sclerenchyma is dead at maturity. And then this is the parenchyma cell. Remember that those are constantly dividing and don't have secondary cell walls.